Hello. My name is Aijing Feng. The topic I'm going to present today is genomic diversity and associate phenotyping of E. coli isolates from the poultry in the southern United States. The avian pathogenic E. coli is one of the most common disease-caused bacteria, which will results in the localized and systematic infections that threatens animal welfare and the productions in domestic poultry. And the antibiotics are used but bring the emergence and the re-emergence of antibiological resistant bacterial strains and the potential untreatable bacteria, which in the end threats both human and animal health. The effective vaccination program is one of the most important strategies in the E. coli disease prevention and control for the alternative to antibiotics, which requires to understand the underlying mechanism of pathogenic E. coli and identifying the genes encoding one or more variants factors of pathogenicity islands and genes that provide bacterial resistance to antibiotics in resistant island is one of the way. And study shows that subset genes that involve different functions instead of a single gene, plasmid, phage, or pathogenicity island could associate with APEC disease. And the project's goal of this study aimed to explore the E. coli isolates genomic diversity with their associated phenotyping of the infection locations. We hypothesized that a subset of genes from the whole genomic sequence associate with disease in different infection locations. And the selected subset of genes would include different functions, and some of them may be PAI and IEI. In this study, 187 E. coli strains were isolated from the sick poultry samples between May 2017 and July 2021. And those isolates cover diversity in the host species, age, and infection locations, including those important infection locations I mentioned. Phylogenetic genetic analysis was conducted using the CV tree and large genetic variation was shown in the whole genomic tree, and the 16 srRNA genes were clustered into two major groups. There are about 5,000 genes were identified by the annotation, and about 1,100 were sharing genes through all the samples, and the rest, about 400 genes were used to 4,000 genes were used to build the gene matrix. One group of the gene matrix described the presence and absence of the genes in the sample using 1 and 0, and the other group of the gene matrix described the gene duplication with the numbers. Infection locations were encoded as one hot matrix. The multitask feature learning model could be used to learn features from this genetic matrix and the phenotype matrix. The model I use for this study is the tree guide multitask sparse feature learning on logistic regression. The numbers of genes features is larger than the numbers of samples, and the numbers of samples in each task is insufficient to train an accurate learner, while the giant learn multiple related tasks so that the knowledge contained in a task can be leveraged by other tasks with the hope to improve the generalization performance of all the tasks. Tree structure was used to group the gene features based on prior knowledge. Regularization is to force some genes weights to be zero during the model learning for feature selections. And the bootstrap resampling is also used to robust feature selections. In the end, we select 27 genes and with the prediction accuracy of 87% in the test set. all the selected genes were in the gene duplication group, and the gene functions were identified using the Python software. Those selected genes include 12 different primary and subprimary and 5 different protein class, as well as the 4 variance factors, and 2 of them are PAI. This study explored the genes associated with different infection locations based on lab collected 187 samples. Public dataset could be added to explore and selected disease-related genomic features, variations within the specific genes, variations in the nucleotide, hypothetical proteins, non-coding regions in the sequence could be considered. And I would like to thank the colleagues from USDA and the Mississippi State University, and also would like to thank the grant support from the USDA. Thank you.